When we look at knowing if evolution and natural selection have occurred, it's one thing to be able to look at a population and notice that there has been change. And here really what we're looking at are the phenotypes, the physical appearances. And we can kind of track that over time. We've got some specific examples of looking at natural selection, looking at evolution. But the best thing in science is when you can quantify it, when you can actually measure it with data. So what we're doing is we're looking at some tools that will help us measure evolutionary change. And so the big question that we're answering is, how do we know if a population is changing over time? That is kind of the definition of evolution. And really, if we want to be even more specific, we would say over generations. So how do we know? Well, one of the ways that we're going to know is we're going to look at the gene pool. And so when we talk about genes, we have to remember that evolution is not acting on the genes. If we're looking at a bird being a selection force, the bird is probably acting on phenotypes, whether it's a behavior or a physical trait. But when the bird selects for phenotypes, we are actually also selecting for genotypes because the phenotype is caused by the genotype, at least in inherited characteristics. And this is why Lamarck's idea of acquired characteristics really doesn't show change over generations. So how do we look at the genotype? Well, one of the things that we are able to do already from our genetic study is look at physical appearance and make predictions about genotypes. Now, keep in mind that you're going to need to be given some sort of data to start with. You might need to be told that one is dominant and one is recessive. You might need to be given a cross and examine the offspring. So, for example, if they were actually crossing these two and this were an offspring, we would know something. If these two mated and had this baby, we would know that white was recessive, and we would know that they were both heterozygote. So they're going to have to give you some sort of data. Now, this picture is really helpful because it puts all of this into a scheme that we already know. We already know genotypes. We already know the idea of gametes. And so this picture is really helpful to, after the notes, go back and really look at again. The main idea is that the gene pool is going to represent all the alleles possible and in what quantity. So when we look at a gene pool, we're actually trying to say, if you are homozygous dominant and you have two dominant alleles, what does that mean? If you are heterozygous and you have a dominant and a recessive, what does that mean? And if you are homozygous recessive. So in this example, they've gone ahead and told us the frequency of the genotypes. Now, if we wanted the frequency of phenotypes, what would we do? We would add these two together. And this would be 0.65 dominant and 0.35 recessive. Now, interestingly enough, when they give us the frequency of each of these, we can determine how many dominant alleles there are and how many recessive alleles there are. And that is this very first equation. So allelic frequency is really just how often or in what quantity each allele occurs. So we're going to use this phrase a lot, allelic frequency. And this is actually what we're going to look to see if it's changing, is the frequency of the allele changing. So how do we determine the allele frequency? Well, we look for how much there is. And P plus Q is an equation that says, if I have two alleles, 100% of the population is those two alleles. So the examples we're going to be doing are dominant recessive. Could you do this with codominant, incomplete dominant, any other gene that has an allele? Yes. But thankfully we're starting out with something really comfortable, which is dominant recessive. And what this means is that P is going to represent our dominant allele. Q is going to represent our recessive. And the 1 means that 100% or 1 frequency means that all of the alleles, the gene pool, is either P or Q. P plus Q equals 1. There are no other alleles. We've got P, we've got Q. And the combination of those two is 1. Well, interestingly, we can do lots of other things with this. We can subtract P from both sides. And we can say that Q equals 1 minus P. Or P equals 1 minus Q. And so this equation is going to be really helpful later. P and Q are the alleles. P being the dominant, Q being the recessive. So you do need to go ahead and memorize that. So what does this mean? This equation is just telling us the frequency of both. But this is really hard to figure out because when I look at an organism, I don't see the letters. I see the phenotype. So it turns out that Hardy-Weinberg have developed a second equation to help explain the phenotypes. P squared is actually going to be, think about this, AA, PP, P squared equals the homozygous dominant genotype. So now I have this equation that's actually just the square of this. Now I have this equation where P squared represents any organisms that are homozygous dominant. 2PQ represents the fact that I could be big A, little a, or I could be little a, big A. Now we've always written this as big A, little a. But remember, you could have gotten this one from mom and this one from dad. Or you could have gotten the recessive allele from mom and the dominant allele from dad. So when we create this equation, we have to account for the fact that there are 2 ways to be heterozygous. And then finally, Q squared is going to equal homozygous recessive. So how does this relate back to this equation that they made? This equation says that these are the 3 genotypes. Those are the only 3 genotypes. And so when I add them together, they equal 100%. So these 3 genotypes, these are the possible genotypes. And they equal 100%. So now the interesting thing is, based on the data that you're given, you can actually solve problems. And we're going to start with kind of an easy one, and then later go into some harder ones. Now when we look at this chart, they have gone ahead and given us a lot of the data, but we can go through and figure out chances, and we can look at this to kind of understand where these equations came from. You do need to memorize P plus Q equals 1, but it makes sense. These are the only 2 alleles. And P squared plus 2PQ plus Q squared equals 1. Again, these are the only genotypes. So let's work a problem, and it'll make a little bit more sense. A population has a percentage of the homozygous recessive genotype is 36%. Using that 36%, calculate the following. The frequency of the AA genotype. Now these are going to deal in decimals, and we're going from a percentage. So we just have to write it as a decimal. So the frequency of the AA genotype is 0.36. Now we're going to need to get into our symbols here. So 0.36 is actually equal to Q squared. 0.36 is Q squared. So if I have what Q squared is, and I want to know the frequency of the A allele, I really just need to take the square root of this. So this means that the frequency of the A allele is going to equal the square root of Q squared, which equals the square root of 0.36, which, careful, equals 0.6. Be careful. And this is Q. Now I know Q. Well, now they want the frequency of the dominant allele. Well, you haven't told me anything about the dominance, except you kind of have. Now be careful, because some people will say, if 36% equals Q squared, they'll say, oh, that means 64% equals P squared. No. 64% actually equals P squared plus 2PQ. 64% is my dominant phenotype, but that's not my dominant homozygous dominant genotype. So be very careful about what you assume. 0.36, you actually almost always want to start with the recessive. 0.36 is Q squared. I take the square root of Q squared, which is the square root of 0.36, I get 0.6, and that's what equals Q. The reason you can't just take the square root of 0.64 to equal A is because 64 does not equal P squared. So to get P squared, we'll get there in a second. To get P squared, we're actually going to go from P. So it turns out that 0.6 equals Q. P is 1 minus Q. So P, which is the dominant allele, equals 0.4. And now I can find out my two genotypes. 0.4 squared, again be careful with your math, is 0.16. And to get this, I'm going to do 2PQ. So now that I have P squared equals 0.16, that's what this is. My heterozygote is 2PQ. So that's 2 times 0.4 times 0.6. This together, 0.6 times 0.4 is 0.24, which is 0.48. Now the best thing I can do is add these together, make sure they make 100. So we'll just check our math. P squared plus 2PQ plus Q squared should equal 1. And I've got 0.16 plus 0.48 plus 0.36. These together equal 0.52 plus 0.41. 48 equals 1. So, definitely a lot of information here. The key thing is that you're able to use logic to figure out how are these equations helpful, and that you don't rush. If you rush, you're going to start taking square roots of things that aren't really what those equations stand for. So the best thing you can do is really go back and by yourself answer, why does this equation make sense? What is P? What is Q? Why does this equation make sense? What are each of these variables representing? And if they give me some data, where can I go from there? There are tons of Hardy-Weinberg. That's what this is called, the Hardy-Weinberg equations. Tons of help on this, so if you Google it, I'm sure you'll find some extra practice problems, but we'll be doing them in class.